Hi, my name is Melissa Mayday, and today I'm going to talk to you about the article Microbiota, Alterations in Amino Acid Deficiency in Pediatric Depression. So to start off, I'm going to talk to you about Major Depressive Disorder, or MDD. So the global impact of major depressive disorder is that 264 million people of all ages are affected by MDD worldwide. This is creating a massive epidemic of mental health illnesses in the world, and it is estimated that 2 to 8% of adolescents in the United States have this disorder. MDD is one of the top leading causes of general disability, which increases the bioburden of disease. Depression in adolescence is often seen with an increased risk for academic failure, substance abuse, social isolation, and even suicide. Early onset of MDD often leads to chronic depression if not properly managed. The key to disease management is early detection and proper treatment. MDD in pediatric population normally co-occurs with anxiety disorders and other psychiatric conditions. This makes early detection very, very important. Raising awareness and reducing cultural stigma is a key step in reducing MDD in adolescents. The overall takeaway is major depressive disorder is a leading cause of disability worldwide, and it affects both physical, mental health, social functioning, and decreases the quality of life. So I'm going to briefly talk about some key characteristics and risk of MDD in adolescents. So as I said, it affects about 2 to 8% of adolescents in the United States. Some symptoms include persistent sadness, loss of interest, impaired social and academic functioning, and is linked to difficulties in school, social withdrawal, and increased risk of substance abuse. 1/2 of MDD patients have co-occurring anxiety. Early onset of MDD in adolescents often causes chronic illness in adulthood. Suicide is a leading cause of death among adolescents with MDD. Lack of treatment may also lead to high risk of suicidal tendencies in the population that does not get treated. So due to these high levels of suicidal tendencies, development of pharmaceutical and just overall solutions to this problem are very important. However, the microbiota is not the only thing at play in developing MDD. There are biological factors such as brain chemistry, genetics, that also play a part. Environment plays a part. It's very multifaceted. So next I'm going to briefly talk about the gut-brain axis. The gut-brain axis is a bidirectional communication system between your gut microbes, the immune system, the nervous system, and the enteric nervous system. And these all talk together by sharing metabolites, and through the vagus nerve, they pass these metabolites back and forth to signal to each other. For example, changing the levels of serotonin or glutamate directly impact the function of the neurotransmitters. And this just shows that there is this communication between all the different parts of your body. Microbial metabolites from the GI tract can cross the gut barrier and affect brain regions. These regions are often involved in mood and cognition, such as the prefrontal cortex. So there are lots of studies that have backed up claims about the brain-gut axis. These studies showed that the gut-brain axis can cause ALS, anxiety, and a whole bunch of different psychiatric disorders in adults. There hasn't been very few studies showing this in adolescents. However, MDD in adolescence is on a rise, so it is very important for us to start studying how the gut microbiota of children is affecting how they are thinking. Previous studies showed children and adolescents with anxiety disorders have an altered gut microbiota. These studies show that there's a pro-inflammatory bacteria that is colonizing over the preferred beneficial bacteria. This increased inflammation changes amino acid metabolism, such as these amino acids include lysine, glutamine, and tryptophan. These all affect brain development, and this was further shown through functional MRIs that showed how amino acid deficiencies reduce functional connectivity in the prefrontal cortex. And just to reiterate, the prefrontal cortex has been connected to mood regulation. Also, there has been multiple studies that have shown that a fecal microbial transplant into mice has caused the mice to develop these neurological disorders such as anxiety-like behaviors, and this just further proves the use of a gut-brain axis. Alright, now I'm going to break down the article. The article is the Microbiota Alterations and Amino Acid Deficiency in Pediatric Depression. So 563 children and adolescents aged 10 to 18 years old were recruited. 256 had NDD. 307 were healthy. The NDD diagnosis was confirmed through DSM-5 criteria by a trained psychiatrist. This is just standard procedure. And then HC, or the healthy population, was screened to make sure they did not have other psychiatric disorders other than MDD. And then they also determined the level of severity of the depression. So this study was approved by the Ethics Committee of the First Affiliated Hospital of Chongqing Medical University. Written informed consent was obtained from both participants and their legal guardians. And participants were free of physical illness, illicit drug use, pregnancy, and they all consented to getting an MRI. So, their blood was taken from all 563 participants at the same time frame via venipuncture, and they were also all stored and sampled the same way. They were also standardized. The fecal samples were collected from 141 participants via sterile and DNA-free devices, and then they were also frozen to prevent contamination, and these samples were collected at home or hospitalized, and then they were also homogenized before storage. Next, I'm going to walk you through some methods. So the first kind of experiment they did was a plasma metabolic profiling methodology. So here, this was conducted on all 563 plasma samples. They used a liquid chromatography mass spectrometry technique, which is a powerful analytic technique combining liquid chromatography, which separates a mixture into individual components, with mass spectrometry. And this mass spectrometry identifies and quantifies these components based on their mass to charge ratio. This is widely used in metabolomics to detect small molecules such as metabolites in this study. They also included a thermoscientific Vanquish UHPLC system and Orbitrap Exploris 480 mass spectrometer. These are both high-performance, high-throughput systems of HPLC and mass spectrometry. They also did a sample separation using HILC, which is a chromatography for polar molecules, and RPLC, which is a chromatography for nonpolar molecules, with defined mobile phases and flow rates. Both techniques allow broad metabolome coverage, improving detection of diverse metabolite classes. This allows for them to get both the polar and the non-polar metabolites, creating a more broad and overview of what's truly going on. The data was then processed via a bunch of different software such as ProteoWizard, MetFlow, XCMS, and it was processed for peak detection, normalization, and metabolite annotation. And then differential metabolites were identified using PCA, which detects clustering based on metabolic profiles. It also used the Wilcoxon-Mann-Whitney test, which compared two independent groups, and it also had an FDR correction. Next, they did a metagenomic sequencing and analysis protocol. So they extracted DNA using the PF MagBind soil DNA kit, followed by quality checks via the NanoDrop 2000. The DNA was then fragmented and sequenced using Illumina Nova Sequence 6000 with pair end reads. It then performed some data cleaning and host contamination removal. These were done through programs such as Bowtie2 and FastP. Clean reads were made de novo using MegaHit, and the open reading frame was made through a program called Prodigal. They also then quantified the number of genes using a program called SOAP2, and then they established unigenes. Unigenes are non-redundant gene sets using the shorter reads. And after establishing these unigenes, they matched them to NCBI databases and KEGG databases. A KEGG database is used to infer metabolic functions. They then calculated the alpha and beta diversity, and then they also identified significantly different microbial taxa using the LEFSE, which stands for Linear Discriminant Analysis Effect Size. Alright, next they took MRI data to determine functional connectivity. MRI data was analyzed from 111 patients with major depressive disorder and 51 healthy people to investigate links between brain function connectivity and blood plasma metabolites. MRI scans were acquired using a Seisman Magnetome Skyra 3.02T scanner. The brain tissue was segmented into 3 regions based off of the cerebrospinal fluid, white matter, and gray matter. Brain images were spatially normalized to a standard brain model, and then the images were corrected for motion, slice time, and distortion. They then performed a functional connectivity analysis, and this was done using a brainotome atlas, which divides the brain into 246 regions. They then created a matrix for each participant's brain of these 246 regions to see the strength of the functional connections between said regions. And then they calculated a family-wise error rate correction using 5,000 permutations of data to determine which connected components were significant, and performed spin-based permutation test to account for different lobe sizes and spatial autocorrelation, which is just like because certain brain regions, when they're close together, they act similar, and it's hard to determine on the MRI which region is truly being used. Alright, they then did a fecal transplant in rats. This study investigated how gut microbiota for MDD patients versus healthy controls affected behavior and brain biochemistry in rats. They used 19 individuals with MDD and 19 healthy controls. They then took 0.8 grams of feces from each donor and homogenized them in saline. They then took 24 specific pathogen-free male Sprague Dawley rats aged 21 days postnatal, and they orally gaved the rats, and they housed them under sterile conditions. This means they autoclaved their food, water, and bedding, and they also filtered the air that they were exposed to. And these rats had a 12-hour light-dark cycle. Before they received their daily gave of fecal transplant, they were exposed to 2 weeks of antibiotics. These antibiotics included ampicillin, metronidazole, neomycin, vancomycin, and amphotericin B. And every single day they got 10 milliliters per gram of body weight for 2 weeks, and then they confirmed the bacterial load via qPCR targeting of the 16S rRNA gene. They then performed behavioral tests. At the same time, these were the sucrose preference test, which assesses their preference for doing things, which is a core symptom of depression. They did the open field test, which measures locomotor activity and anxiety, an elevated plus maze, which evaluates anxiety-like behavior, a forced swim test, which quantifies depressive-like behavior, and then they did a brain dissection of the prefrontal cortex, where they then took this brain matter and did a liquid chromatography mass spectrometry to identify the amino acids that are there, and then they also compared this to NCBI and KEGG databases. The last thing they did was a dietary lysine restriction, which examines how dietary lysine influences behavior, gene expression, and protein levels. In rats exposed to chronic, unpredictable, mild stress, which is a validated model of depression. So they suspected 48 SPF Sprague Dawley rats with the same conditions, environmental conditions as the fecal transplant rats, with the autoclaving and the filtered air and the light-dark cycle. They then split these rats up into 4 groups. A control group that had 100% lysine food. There was a control group that only had 70% lysine diet, and then they had Cums rat with 100% lysine diet, and a Cums rats with 70% lysine diet for 4 weeks. And then twice a day they were exposed to stressors to show that they to induce depressive-like behaviors. And then they performed all the same behavioral tests that I talked about in the previous slide. Alright, figure 1 is the amino acid depletion and MDD association. So panel A is a partial least square discriminant analysis, which visualizes separation between metabolomic profiles of MDD patients and healthy controls. So in panel A, you can see clear separation, indicating distinct plasma metabolic signatures between the 2 groups. Panel B is a volcano plot of differential metabolites. The y-axis shows statistical significance, while the x-axis shows magnitude of change, and this is for 1,300 metabolites. So red means they were upregulated in MDD, blue means they were downregulated in MDD, and gray was not significant. So in B, you can see most significant altered metabolites in MDD were downregulated. In panel C was a KEGG level 2 pathway enrichment analysis, and this is a mapping of 360 differently expressed metabolites in the KEGG database. And in panel C, you can see that amino acid metabolism was the most enriched pathway. In panel D, is also a KEGG level 3 pathway, which specifically shows amino acid metabolisms of 13 different amino acids, including arginine, proline, tyrosine, and lysine biosynthesis and metabolism. Red shows metabolites increased in MDD, blue is decreased in MDD, and you can see that most amino acid related pathways are suppressed in MDD. Alright, and panel E is a pathway of 13 dysregulated amino acids, and it just shows the key amino acid metabolite pathways, and it's showing that lysine, methionine, tryptophan, isoleucine, leucine, phenylalanine, valine, and histidine are all essential. And then it goes on to show different metabolites and what their precursors are and whether or not they were downregulated or upregulated. And the overall conclusion from panel E was that these changes reflect widespread impairment in neurotransmitter and energy-related metabolism. Alright, figure 2 is the gut microbial composition changes in MDD. So your panel A is your alpha diversity, which the overall summary of panel A is that it wasn't changed. Alpha diversity measures the diversity within a single sample, reflecting richness and evenness. And it used the Shannon, Simpson, and Chow 1 indexes. As I said, there was no significant change between any of these. Panel B shows the beta diversity, which compares microbial composition between samples, and this used the PCOA analysis. And it showed that there was statistically significant separation between MDD and HC groups. Panel C was a microbial co-occurrence networks are altered, and it shows that co-occurrence networks show which microbes tend to appear together or influence each other. And it shows that when you have a healthy person, they are more densely connected than in MDD. Panel D is a network topology which confirms reduced connectivity in MDD. It shows that they have lower MDD networks have lower node degrees but higher natural connectivity. Node degree is defined as number of connections each microbe has, and natural connectivity is how robust the network is to losing nodes. Alright, and panel E shows that nodes are removed in order of importance to stimulate stress, and in MDD, the networks are maintaining connectivity longer, suggesting greater resistance to targeted disturbance. Panel F shows that there is an increase in pro-inflammatory taxa such as Agarthalake bacteria, and there is a decrease in beneficial species such as Bifidobacterium. Panel G shows the functional impact on amino acid pathways, and there's an abundance of 6 microbial KEG modules involved in amino acid metabolism, and there's alterations in all of these in MDD associated microbes. Figure 3 is the MDD donor microbiota induces depression in rats. So Panel A is just the experimental design, and panel B is the sucrose preference test, which shows reduced sucrose preference in rats that received MDD donor microbiota, which indicates anhedonia. Panel C is the open field test, which panel C shows decreased exploratory behavior and increased anxiety in MDD rats. Panel D is your elevated plus maze. This test measures the time spent in open and closed arms, and the MDD rats spent less time in open arms, indicating heightened anxiety-like behavior. Panel E is your forced swim test. This assesses immobility time as a proxy for behavioral despair, and MDD rats showed increased immobility, therefore increased depressive-like behavior. And then panel 5 shows that MDD rats have reduced levels of essential amino acids. Panel G is a heat map or scatter plot linking microbial taxa and behavioral outcomes, and shows that MDD-associated microbes correlate with worse behavioral and metabolic outcomes. Alright, figure 4 shows the amino acid deficits in adolescent depression. So at panel A analyzes 29 altered functional connections among 27 brain regions that differ in MDD. This shows that there's a reduced functional connectivity in MDD. Panel B shows that there is decreased functional connection in MDD. Panel C shows there are 276 metabolites that are directly associated with functional connectivity. It also shows that 72 of them also have differential abundance in MDD. Panel D ranks metabolite classes by how many are both associated with brain function and altered MDD. And it shows that the top metabolic class was amino acids. Panel E shows the lobar level analysis of amino acid associated functional connectivity. Panel F is the node level and region specific map of where AA linked functional connectivities are concentrated. Alright, and then figure 5 is the causal link between microbiota and behavior. So panel A is the overall experimental design, which I already discussed. Panel B shows on the left panel of panel B is the sucrose preference test, which shows that MDD rats showed reduced sucrose preference, indicating anhedonia. Right is the forced swim test, which shows that MDD rats have increased immobility, indicating depression-like behavior. Panel C shows targeted metabolomics, measured 20 amino acids in the prefrontal cortex, and that 14 of these amino acids were reduced in MDD rats. And panel D shows that many downregulated amino acids in MDD patient plasma were also downregulated in rat prefrontal cortex after FMT. Alright, next is figure 6, which is the lysine restriction experiment. So panel A, again, is just the experimental design. Panel B is the behavioral test. The sucrose test on the left shows that the stressed rats with the 70% lysine diet had the lowest sucrose preference. They also had the highest immobility time in the forced swim test, which is the right side of panel B. In panel C, they took the RNA from the prefrontal cortex and saw that there were 1,428 differently expressed genes in the stressed rats with the 70% lysine restricted diet. And then panel D shows that a KEGG enrichment analysis shows that glutamatergic synapse pathways was significantly altered by lysine restrictions in the CUMS rats. This pathway includes genes for EAA2T and EAA-T3, which are crucial for glutamate uptake and synaptic balance. Panel E just is a Western blot analysis that confirmed that the AAA2T and T3 proteins were significantly reduced in the MDD rats. This study provides a holistic exploration of the microbiota gut-brain axis in MDD. So the key findings were that children and adolescents with MDD show significant depletion of amino acids such as lysine, methionine, tryptophan, phenylalanine, and glutamine. These amino acids are crucial for neurotransmitter synthesis, brain growth, and synaptic function, especially during adolescence when nutritional demands are high. There was also an altered gut microbiota composition in people with MDD. They had a distinct microbial profile, including increased pro-inflammatory bacteria and decreased beneficial bacteria such as bifidobacterium. There was functional analysis revealed disruption in microbial amino acid metabolism pathways, specifically with aromatic amino acids and arginine and proline metabolism. These changes potentially impair microbial contributions to systemic amino acid availability. The plasmid amino acids were also found to correlate with brain connectivity. Functional connectivity deficits in the prefrontal cortex were associated with plasmid amino acid levels. The frontal, temporal, and subcortical networks were the most affected, consistent with regions involved in mood regulation. And FMT from major depressive disorder patients induces depressive phenotypes in the rats, and they had increased depressive-like behaviors such as anhedonia and despair, and they also had a significant depletion of amino acids in the prefrontal cortex, mirroring human plasma findings. And then there was also the finding that lysine restriction increases depression susceptibility. Adolescent rats fed with lysine-restricted diet under chronic stress displayed more severe depressive-like behavior and showed downregulated expression of EAA, T2, and T3 proteins in the prefrontal cortex, and these are key transporters that maintain glutamate homeostasis. This suggests that lysine deficiency may impair glutamatergic synaptic function, contributing to major depressive disorder. So there were some limitations of this study, such as they only used male rats, and they also only took samples from Chinese children, since the study took place in China. So it'd be interesting to see how a westernized diet would correlate to this. So in conclusion, children and adolescents with major depressive disorder have significant deficiencies in plasma amino acids. These amino acids are linked to gut microbiota changes that disrupt amino acid metabolism. The fMRI shows that these deficiencies correlate with the functional connectivity of the prefrontal cortex. Additionally, fecal microbiota transplantation from MDD patients and dietary lysine restrictions both caused CUMS rats to develop depressive-like behaviors, further validating the study. And these are some questions that I have. For example, just one of them is how might interventions targeting gut microbiotas be used to restore amino acid balance and improve depressive symptoms in adolescents, and what challenges would arise in translating this into clinical practice. And those are some additional references. Thank you.